So we are in the watermelon field in Plant City, Florida. It's 8:30 in the morning. So we're here to collect a leaf sample for the sap analysis. So we are going to collect young new leaf as well as old leaf for the sap analysis, and we'll make sure I have 80 grams for each sample, and I'll show you how it's done. Watermelon leaves are not very hard to collect the sample from, but I just brought the clipper just because it's easy. So if you look at this watermelon vines, they are kind of all together. It's just even hard to see which plant is from which bed, but I'll try to make sure I collect the sample from the leaf right plant. So here, if you look at this, this is very young leaf. Yes, you don't want to collect a leaf sample like this. It's very young. You want something that's recently matured, which is from here, it doesn't seem like it's increasing any, it just looks any different. So I think these are recently mature leaf. Then again, some of the leaf, if you look at more close to the plant, this one looks older leaf. So we'll collect the sample from this older leaf as well as from this young leaves. And for this, we are not going to take a whole petiole like sample, so we'll just clip the leaf from here, and this will be our one leaf sample. So we'll collect enough number of leaf that will give us 80 grams. So during the sampling, when you're looking for the older leaf, you go more towards the base of the plant, and you will see these older leaf, versus if you're looking for more younger leaf, you will have to go close towards these tendrils. They have new leaf forming, so these will be a new young leaf. Now let's see how much this leaf weighs. So this looks like a 60 gram, so you want a little bit more of these old leaves, and these are young leaf. This is only 22 grams. Definitely we need more younger leaf to get that desired weight. After you collect the leaf sample, you want to put in the paper towel and dry it out, because it may have some dew, some water. You want it like a dry leaf sample, so use a paper towel to take away the extra moisture before you put in the bag. So sampling small leaf can be a little tricky because it weighs it doesn't weigh much, so you have to collect so many leaves. So that's why make sure to bring big bags, because this is a one sample for a small leaf that I couldn't fit in a one bag, so I have to use two different bags. So make sure to bring a big leaf, and also the small leaf samples are more sensitive. They can get squeezed or damaged, so make sure be more careful when you're handling a young leaf. Old leaf, they're pretty sturdy, and they can collect a few numbers and it will reach the weight you desire. So we are done with the sampling. It took a while, especially if you're collecting multiple samples. So we're going to directly ship now. So otherwise I would put in the cooler and stuff, because I'm just shipping right now. I'm just gonna go and ship it to the lab. But if you're not going to, you have to put it in the cooler and keep it cold, but not freeze them. So this is the watermelon sample we collected yesterday. I couldn't send it yesterday, so I put it in the refrigerator. And I finally got a label, because this is going to a Netherlands to a Nova crop control. So I got a label with a name of the plant and what growth stage leaf is, what is we going to do for it, and I put the date on it. So I got this label for each sample, and I also tagged each sample with this is untreated, the young leaf. And then if it's a untreated young leaf, then I'm going to use let's see, untreated young leaf. So I'm going to put this inside of the Ziploc bag, and also a tag for this, so that we know what is this sample. And after we do that, we put this inside. We put this inside, and we zip lock it. Like I said, the sample needs to be dry. Try to take the air out so that it's less volume. After I put all tag and label inside, I put it inside the paper bag that I got from Publix. This tag acts as insulator with the ice that we're gonna put inside this ice bag. So this is the ice bag I got from Publix because it's gonna travel across the world and it's gonna be there for a few days, and it's the summer, so I like to use this. Put inside, and then I will put some ice around it, and I will lock it very tight, and I will drop it at the FedEx today for this to be mail-out. Now we have the result. This is just a sample. I just want to show you how the result looks like. We're not going to talk in detail about or analyze what nutrient status we got from those leaf samples we collected. I just want to show you what you can find on the result. So looking at here, this is just the name of the lab that did the test in Netherlands. This is a sampling date, location, what cultivation, what crop, what plant pot. We submitted a leaf, young leaf, and the old leaf. The one is young, two is old. And there's different categories: deficient, low, optimum, high, excessive. And this is for young one, and this is for old leaf, for different kind of minerals and nutrients. And this is the numerical value of the concentration, and this is what optimum should look like. So one thing you get with the SAP analysis is a sugar, total sugar content that you don't get the regular tissue analysis. It gives you that total sugar content on the SAP. Some of the nutrients you can look at, like potassium. You can differentiate what's going on in the young and the old leaf, because potassium is a mobile nutrient. That's why if you look at here, you have the same amount of potassium, and that means if there is a lack of potassium in the younger leaf, it moves from older to the new leaf. So there's you don't see like a potassium deficient younger leaf when there's a potassium on the older leaves, because it moves around. But on the other side, calcium is immobile nutrient, it doesn't move. So when you look at here, you see this is the older leaf that has an optimal amount of calcium, versus younger leaf has a very deficient in calcium. That means it isn't moving from older leaf to the younger leaf, so it is a immobile nutrient. So that kind of thing I want to show here. It has all other nutrients like magnesium, nitrate, total nitrogen, ammonium nitrate, different kind of things. You get this other micronutrient that you don't generally get with the dry tissue test. Is a silicon. I have not really found you have to pay extra for that, but it comes with the SAP analysis. So this is just an example how it looks like, and I hope that will help for you to analyze your results.